Okay, hi guys, we're going to be discussing now genetic code, further continuing with the molecular biochemistry. Previously, we discussed the basics of DNA, and all this was basically discussed under it. And now, genetic code and its features, what makes it so exceptional. So, the first feature of genetic code will be that it is very unambiguous, okay? Unambiguous. Sorry, unambiguous. So what does this mean? It's basically when we say that the genetic code is very unique or very exceptional. Why? Because genetic code is crucial in what? And the main thing that it does is that it has to form protein. So DNA to RNA, and then mainly the protein that has a part to play in a human body, right? So this protein, how it is so controlled that this protein synthesis can occur from the DNA coding system. So this coding or genetic coding system, how it is so ambiguous, that is that there are in this table, this short table that I've just posted here as an image, it shows that there are like 3 bases for each amino acid. So these are the amino acid. We have phenylalanine, leucine, serine, and all the other amino acids, and each amino acid is coded by a triple code. This triple code is a codon. Now, unambiguous or an ambiguity of a genetic code means that each codon is coding for a specific amino acid, or only amino acid basically. So this makes it so specific that each codon, for example, phenylalanine, UUU. Now this codon means a triple code, right? So this codon specifically is going to be only coding for whenever this codon comes on a DNA codon system, where the tRNA comes and start coding. This pattern is only for phenylalanine. And for example, if it is UUA, then this is specific for only leucine, nothing else. This is specifying it, right? So this is the basically an ambiguity example of what it means. Second feature of genetic code is that it is redundant. It is redundant, or we also call it degenerate, right? This is like to remember this is a specific name for the feature. Redundancy means that when we discuss that each codon is coding for a specific amino acid, means codon is single for a single amino acid, but each amino acid is coded by multiple codons. So it's simple basically. Phenylalanine, you can see, is coded by 2 different codons, means that UUU is only going to code for whenever we see UUU, we know it is phenylalanine. Okay, so when we're discussing phenylalanine, we say that it has 2 codons, possible codons, which is UUU and UUC that we see in the table here. So this is basically when it comes to discussing redundancy of the genetic code, right? And this is very important actually. This pattern, or this multiple codons coding for a single amino acid, basically allows for or prevents the protein from mutations. Basically, when the tRNA comes and it start coding, the first 2 positions, UU for example, UU, the first 2 positions are very strict, that it has to be coded the right way. But the third position here can vary. tRNA is more relaxed in its way to basically code for this third position, so we call this as a vowel position. Now vowel position is a third position. Vowel position is a third position on the codon that can vary, that has basically the flexibility, or it is less strict in, in basically transcribing. So if it is flexible, then it might have, there can be a point mutation. There can be a prevention from point mutation, that even if it is UU, for example, if this third position changes from U to C, then it's still phenylalanine. So we don't have to worry about, that any, possible mutation or change of a protein, right? So this basically allows the amino acids to ultimately result in the same protein with this coding system, that each amino acid is coded by multiple codons. So this is actually a benefit for preventing any point mutations, right? Then we also need to know that the genetic code is very universal, right? Means it is preserved throughout life, right? Preserved throughout life. It's universal. It's like we discussed, 75% is methylated. It's preserved. It's controlled protein mutations, protein synthesis already going on, and it's unlike any DNA that has high variability. So there is less rate of variability. That's also a very exceptional characteristic of genetic code. And furthermore, it is also commaless and overlapping, non-overlapping. Why? Because definitely it has a fixed starting codon when it comes to protein synthesis, and unlike the the three basically stop codons. So it has a fixed starting codon, which differentiates it from multiple, for example, viral DNAs that have multiple starting codons that are more prone to mutations. So this is more of a non-overlapping, so it always start from UAUG. This is a codon to remember, basically. UAUG, that is coding for methionine. So this is the amino acid needs to remember that it will always start with met. Okay, AUG. This is the codon to remember, and which basically makes the genetic code so exceptional, so non-overlapping. So these are the characteristics that we discussed here. Okay, it is unambiguous. That means each codon is specific for amino acid, and how it is, we saw it in this table. And then it is redundant and degenerate, which means that each amino acid can be coded by multiple codons, which basically prevents the point mutations, or even if the mutation occurs, like on the third position, which is a wobble position, that allows flexibility, and it can usually be silent mutation without affecting the protein synthesis itself. Then it is universal, preserved throughout like life of the person, and it is also on non-overlapping comma less. Now we're going to further go with DNA methylation. Maybe we will be covering half of it. DNA methylation. Now DNA transcription and translation is highly preserved. You just don't have to say that the DNA in the nucleus is just all of it just transcribing and translating all the time, and the cell is dividing, right? This is more of a complex thing going on, right? Because when we say that it's 70 to 75% is preserved. And why it is preserved? Because of methylation. What is DNA methylation, basically? DNA methylation is that there is a methyl group, right? From its very basic meaning, that there's a methyl group, which is CH3. Okay, this very group is going to attach to a base. Methyl group attaches base. And when it does that, it makes the bond of bases stronger, right? It makes the bond of the bases stronger. Now, what it attaches to in DNA methylation specifically is CpG islands. CpG islands is cytosine to guanine. And as we have discussed, there are three bonds between cytosine and guanine. It's a stronger bond. Now what happens is that the methyl group CH3 is going to attach to cytosine, the pyrimidine, with a single ring, okay? And when it attaches to cytosine structure, it makes this bond stronger. So all the areas where there are CpG islands, okay, CG, DNA methylation is basically preserving those areas, right? DNA methylation is going to be preserving that area, and most of it will be preserved, and there will be a methylated DNA, thus preventing it from transcription. One thing again is that when there is methylated DNA, there is less transcription going on in that DNA, or basically it inactivates transcription, right? So what is DNA methylation? It's basically a way of preserving most of the DNA. It's a heritable epigenetic modification that regulates gene expression by adding a methyl group to a DNA molecule without altering its underlying sequence. So without the alteration, the methyl group basically comes and preserves this DNA. This process is crucial for normal development and health. And basically, to go further in with the process, is that the cytosine bases, the dinucleotides that we discussed, cytosine guanine bonds are becoming stronger here. So the enzyme that is involved to remember here is the DNA methyltransferases. By this process, why this catalyzation, it is undergoing methylation transferases, right? In short, we say DNMTs. These are the main enzyme that are responsible for DNA methylation, right? So what happens is that this methylated DNA basically attracts the methyl CpG binding domain proteins, and these these proteins are going to recruit other protein complexes, which are histone deacetylases, which further will condense the chromatin into a compact inactive form, heterochromatin, that prevents transcription. So when we say how it basically inactivates transcription, is simply to remember that it helps the DNA DNA part of DNA to basically form into a heterochromatin that we discuss as a more compact and condensed form of chromatin, right? It attracts histone deacetylation, which is also preserving the DNA, and these deacetylases enzymes are going to thus result in inactivation of the transcription and making the DNA more condensed and compact and preserved into a heterochromatin, basically. So this is one thing to remember. Then furthermore, we also will see that how it is important in preventing cancer, because the cancerous DNA will be highly unmethylated, which will be easily recognized by the immune cells, and it will be easily targeted and eliminated. So for cancer, when we say cancer, and also the bacteria, and also autoimmunity, that's a different case. We'll take it afterwards. For cancer and bacteria specifically, we say its DNA is unmethylated, highly unmethylated, so there is increased transcription going on, there is mutation going on, it's easily basically attracting the immune reactivity, so there is increased immune reactivity for it, right? So this will help the body to basically eliminate those specific bacteria cells or cancer cells, right? This is important in that. And other key functions in healthy individuals, that why DNA methylation is important for maintaining the health as well, is that it's important for gene regulation, right? Let me just write it. It's important for gene regulation. It's important for embryonic development. It's important for genomic imprinting. It's important for X chromosome inactivation in case of determining gender. It's important for suppression of transposons, basically, right? Which are basically jumping genes, we say. Gene regulation, how it is important is that is for controlling the gene expression and helping to turn the genes on and off during the self differentiation. So it is basically regulating the gene sequencing or gene expression during cell division. Embryonic development is that it helps maintaining it basically helps maintaining the crucial DNA, and again the preservation part that comes in for tissue and organ formation during embryonic development. Then genomic imprinting, when we discuss this, that a specific allele has to be expressed, or specific parental allele has to be silenced. So DNA methylation plays a crucial part in it. It relies on the imprinting, basically relies on methylation to silence one of the parental alleles, so only the other is expressed, right? So genomic imprinting, that for example in disease control or all that, for silencing any genes, methylation is playing a crucial role in there. X chromosome inactivation, like in females, like one of the two X chromosomes, we have XX, right? So one of the X chromosomes are going to be silenced to balance the gene dosage with the males, so that's also having some impact of methylation. Then control of these jumping genes is that it basically, there can be gene there can be differences or variability going on with the genes and sequencing, so these jumping genes can be easily silenced with this methylation, it preventing ultimately limitations, right? So yeah, this is mainly the DNA methylation. To have an overview, DNA methylation basically preserves the DNA in form when the DNA has to transcribe and translation. The DNA transcription and translation in itself is a highly preserved. Why do we call it preserved? Because there is DNA methylation that preserves it, that 70-75% of the DNA is preserved. DNA methylation is basically only the methyl group via the enzyme DNMTs are attaching to the base or nitrogenous base of the CpG island. CpG islands is cytosine 1 islands or bonds that make these bonds stronger. The methyl group, when it attaches, it makes the bond further stronger, thus making it more stronger and preserved, and ultimately inactivating the transcription and forming the DNA structure to be preserved in a form of heterochromatin, which is more of a compact form of chromatin that disallow any further communication with the transcription factors. And then how it is very important in cancer control or bacterial infection control is that the DNA of cancer cells or bacterial cells are unmethylated, which basically allows more immunity to occur in this case, immune reactivity to increase in this case, and for autoimmunity like SLE, the T cells itself has unmethylated DNA, which is basically the body's own cells that reacts with basically becomes the DNA becomes prone to methylation, and that results in its own destruction. Then it's healthy point of view why it is important DNA methylation overall. We discuss some points here, some things to remember, and that's it. Thank you for watching.